Good morning. Today I'm going to talk a little bit about early detection and early screening measures for cancer. The futuristic part, what's coming. It's very clear that prevention is better than cure. And to prevent disease, we have to develop better and better technology to diagnose even the footprints, the perturbations that are caused in networks in the body by diseases. And I predict a total revolution in the healthcare system in the coming decades, where every disease will be handled in the same way, not just cancer. So let's talk about cancer. So far, screening measures have been rather gross, using pap smears, using colonoscopies, using mammography, using MRIs and CAT scans. But these can only be done periodically. We need to develop techniques which constantly monitor the body for the appearance of the first cancer cells. And to do this, we have to understand the biology of cancer. So without rigorous scientific experimentation, none of it will be possible. And that is why I keep saying over and over again that in order to find the first cell, we have to continue to study even how to deal with the last cell, because all of that will eventually help us trace the steps of cancer back to its origin, its inception, where we can manage to nip it in the bud. So for example, when cancer begins, one of the characteristics of cancer is that it divides faster than its normal counterparts. Now, in order to divide and replenish itself, it needs nutrition. So it starts attracting blood vessels and making blood vessels, new ones by itself, as it gets more and more organized. Whether it's solid tumor or liquid tumor, increase in blood supply is being documented for everything. As soon as blood starts going to an area, that area becomes hot. How hot it can be picked up by scans, by imaging techniques. So why not take advantage of this? A lot of people have. I mean, one of the best examples is the Sanjeev Sam Gambhir lab at the Canary Center for Early Detection of Cancer in Stanford University. Sam Gambhir is leading this charge in developing the futuristic imaging devices that can detect cancer. How? For example, they are working on developing a smart bra. This bra is set up with thermotactile sensors which can detect changes in temperature. And women just have to wear it for a few hours once a week, and it will start recording if there is a change in temperature due to increased blood supply somewhere in the breasts, and alert. I'm not saying we should immediately jump in and take the breast off on that woman. Of course not. The idea is that once one device detects something abnormal, it has to be followed by first constant monitoring over the next several weeks or months, and then if it persists or seems to be increasing in size, then do 10 other marker studies to make sure that this is real and malignant. And how lethal is it? It has to be. So what do we do next? Well, then one possibility is that cancer cells upon formation also start shedding proteins, bits of DNA into the blood. So we start scanning the blood, liquid biopsies for the presence of mutated DNA that only comes from a cancer. And there are certain mutations very typical of breast cancer. So if that confirms, a liquid biopsy confirms the smart bra finding, now we have two things saying the same thing. But some cancers are not that good at shedding markers. So Sam Gambhir has come up with an idea that you can literally yell at a tumor. How do you yell at a tumor? Well, if you think that there is something that seems to be forming in an area which looks hot suddenly, then you can simply point some sound waves, ultrasound waves on it with a very benign sonogram. And as soon as sound waves hit this, it will shake up the tumor, and that will make it yell back by pouring out many of its markers, proteins, antigens into the blood. Not just that, but even whole cells will come out into the blood. And so as you're yelling at the tumor, it's yelling back at you, you start getting the biomarkers collected from the blood. So that's another way. The lab of Bert Vogelstein at Johns Hopkins is another one leading this effort, and is very well supported by the National Cancer Institute. They have come up with a measurement or technique that they are calling Cancer-Seq, and that looks for proteins and genetic mutations that are very typical of some of the most common cancers, for which there were no screening devices present before. For example, ovarian cancer, the screening devices have not been very helpful, but they have come up with better markers. With this Cancer-Seq, the Vogelstein group has been able to pick up early cancers from 70% of the samples submitted to them. So that's another way. What is our effort in this area? We are working with the bioengineers at Columbia University, and Professor Sam Sia, a brilliant young bioengineer and a leading scientist, is our partner. And what the Sia lab has already developed, which has become FDA approved through a company, OPCO, is something called M-chip. It's a tiny little chip which uses microfluidics to take one drop of blood that you can place on this chip. It will travel through microfluidics and interact with antibodies or markers placed at the other end. And so this is, they're placing the measurement for PSA right now. So with this chip, you can be at home and simply every day you can test your PSA level. But if we have a early marker for leukemia, we can put that there. Or we can put leukemia, prostate cancer, multiple markers, and this would interact with all of those. So we are very excited about working with Sam on this further development of M-chip for use in liquid tumors as well. He's also working on implantable devices, which is my dream. My dream is that we should be able to have an implantable device which we can place under the skin that, for example, has a filter built in. And this filter will allow blood cells to pass through, but anything bigger than a blood cell will be trapped on the filter, and cancer cells are bigger. So if cancer cells are being shed somewhere, they will be stopped at this filter and they'll start collecting there. And we can have a scanning device from over the skin every day. You could have this device at home built as an app in your telephone, for example, which will be able to detect if there are more than, let's say, 10,000 cells that have been trapped suddenly over a period of a week. That could be abnormal. So my dream is that we should have these implantable devices developed in the next 5 to 10 years that we can start inserting under the skin in infants at birth, so that we start monitoring right from the beginning of life. And this would become, given the economies to scale and how rapidly a capitalist society takes these things and runs away with it, I think it would be fantastic, just become like immunization. This is another thing that you get inserted under the skin. So there are all these amazing futuristic scanning devices that are appearing on the horizon. My pet peeve is that too few people are doing this. A lot more resources from funding agencies have to be directed in that area as well. And I'm not saying that current cancer research should stop. Absolutely not. We have so many patients to treat. We owe them. We must continue that research, and the rigor of science has to be there. So yes, but at the same time, we should stop some frivolous studies that are being done, like drug development in animal models, a complete waste of resources, time, money, and intellect. We should take all of those resources and invest them and make them into challenging, competitive grant proposals that we want people, younger people especially, to come up with, and fund them for finding better ways of diagnosing cancer early, developing these devices, these techniques, whether they are imaging or ultrasonic or MRI or PET scan based, for early detection. This will be the wave of the future, and this is where I think we really need to invest our resources more intelligently, to go from finding the last cancer cell and constantly chasing after trying to kill that. It is high time that we understand that we have not used all our resources wisely. Let's be humble about it, and let's try to correct ourselves by now focusing on preventive part of the disease as well. Thank you.